Transcription of prokaryotes. Now, it is the creation of a complementary strand of RNA from a DNA template. Here we're going to label. We have our DNA strand. So we have our DNA. This is our coding strand. All right, we're going to label it 3' to 5'. There are 3 stages to the transcription of prokaryotes: initiation, elongation, and termination. First step, we have initiation. We begin by recognizing our promoter site. Now, the promoter site is just a section of the DNA that tells the RNA polymerase where to begin. So we have our promoter site. And I'll just put in parentheses, starting point. All right, now I did mention about the RNA polymerase, and it is an enzyme that synthesizes mRNA. So I'll draw. Our RNA polymerase. So it finds the promoter site and then moves down the DNA coding strand to synthesize messenger RNA. All right. So 1, initiation, little recap. We have our promoter site that is our starting point where our messenger RNA begins to synthesize. Then we have our elongation, and that is the process of synthesizing our messenger RNA. So 2 is elongation. So let's go in a little deeper on elongation. We know that is the synthesis of messenger RNA, but let's give a little more information on it. So in order to make messenger RNA, you need nucleotides, and nucleotides are picked up by RNA polymerase. Now these are RNA nucleotides that consist of bases such as we have our A, G, our U, and C. So we have adenine for A, G, guanine, and U, uracil, and C, cytosine. Oops. All right, so our RNA polymerase picks up RNA nucleotides that are complement to the DNA coding strand. Now, eventually, the RNA polymerase would have to stop, and there is a point at which it does. Once it hits the terminator site, there is an end to the synthesizing into the synthesis of messenger RNA. and this is called 3 termination. Then our messenger RNA is free for translation. Okay, let's do a quick recap. So 1 here, we have initiation, the initiation stage. This is where the RNA polymerase finds the promoter site to begin the synthesis of messenger RNA. It moves along the DNA coding strand to synthesize messenger RNA. This stage is elongation. Now, the RNA polymerase will then pick up RNA nucleotides. Now, adenine, guanine, uracil, and cytosine. These are the bases in RNA nucleotides that are picked up to create messenger RNA. Now, eventually, RNA polymerase will have to stop, and this will take us to 3, our termination stage. The RNA polymerase will then reach the termination site, which is a specific code, a specific sequence of DNA that tells RNA polymerase to stop synthesizing messenger RNA. And then the messenger RNA will go off into the translation stage. Need more help? Click the link in my description box to get started with your microbiology study module.